It's my great pleasure to introduce our Max Planck director colleague Stefan Hell from the Max Planck Institute for Multidisciplinary Natural Sciences. I think that covers everything. And Stefan received a Nobel Prize in Chemistry actually in 2014 together with Eric Betzig and W. Murner for the development of super-resolution fluorescence microscopy. And again, I'm not going to say anything more and hand over the stage to Stefan.
Thank you very much, Jon. It's a great pleasure to be here, and of course, congratulations on this inauguration of the center. So I think it's something that is very special in this country, on this continent, in fact. And I very much look forward to seeing great results in continuation, I should say. I'm going to speak about a very simple problem, which I think has a number of implications in the end for basic medicine, if you want, and definitely for biophysics. And I will start out with a very, very simple question, which is a kind of physics nerd question. How to localize, meaning how to find out the position of a fluorescence emitter, of a single fluorescence emitter of a molecule with maximum accuracy, precision, and I should add, speed. Now, what people did actually in the past, I would say since the 60s, as a matter of fact, they kind of conceived a way of localizing individual molecules in the focal plane of a fluorescence microscope that works like this. You have a wide field epifluorescence illumination, so you have a flat field illumination, and there is a molecule somewhere, for example here, and this molecule, we need to find out where the molecule is. And the way it works usually is to collect the fluorescence that is produced by this individual molecule by the same objective lens, and then the objective lens projects the fluorescence that is emitted by this fluorescent molecule onto a camera. Now, as you certainly know, the net result is a blob of diffracted fluorescence light on the camera, which can be actually visualized here on a computer screen. And then localization means that, of course, we know that the position of the molecule in the focal plane must kind of coincide in space with the position of the maximum of the diffraction fluorescence blob. So in other words, once we have found out where the maximum of this diffraction blob is, then we know where the molecule is, because that position must actually stringently correlate with the position in sample space by simple geometrical optics. Now, the precision with which you can find out the position of this molecule scales inversely with the square root of the number of detected photons on a camera. Actually, the first source that I know of that kind of related this idea is Werner Heisenberg in his booklet written in 1930 when he was on a sabbatical at the University of Chicago. And of course, this kind of relationship was used through many decades in many biophysical experiments, and certainly also in super-resolution microscopy methods called PALM, STORM, PAINT, and so on. Now, the gist of it is that you need to maximize the number of fluorescence detection on a camera. It's very clear. The higher n is, the better localization precision, the better you find out the position of the maximum, the better you find out the position of the fluorescent molecule. But if you need to maximize the number of fluorescent detections on a camera, you have a problem because you need to wait. If you need to wait, that's bad because it's slow. The molecule may move away, and you may get only, say, 100 or maybe 1,000 photons, but not the 10,000s, perhaps, that you need to get a very high accuracy. So you are inherently limited by the photon rate, and you are inherently limited by the detection time. But that's not the only problem. Simple problem is the fact that fluorescent molecules, as you may appreciate, they tend to bleach. Not only they bleach, they also blink. Blinking means they go off for a while, then they are silent, they don't emit. And of course, during this period, the molecule may move away, and then you cannot find out its position. But apart from that, there's another problem that you will appreciate, especially if you're trained as a physicist or a physical chemist. Then you will realize, of course, that a molecular emission or absorption is in fact a dipole transition, meaning that the orientation of the molecule in sample space really matters. In other words, if the molecule emits and it's, say, oriented like this, then it cannot emit in this direction or in that direction. It can only emit in this direction to the side and here. That means that the pattern, as a matter of fact, if the molecule is not freely rotating, but just kind of hindered in its rotation, the pattern of the diffraction blob on the camera will be shifted to the side. That phenomenon can be really big. As a matter of fact, if the molecule is slightly off the focal plane, it can easily amount to 10, 20, 50, or even 100 nanometers. So you might think that you know where the molecule is, and you might get a very high precision of the position of the molecule, but it may be simply inaccurate. You may be wrong about its actual position. Now, the bleaching problem, you can come up with a solution with. And one of the solutions was actually pioneered here in a group by Wahid. You go to low temperatures, you inhibit photo-bleaching, and then, of course, you get high number of detected photons, and you get very, very, very high precision. I decided to take a different approach, and this approach is radically different in a way that it does not rely on primarily on the photons that are emitted by the fluorescent molecule, but to a large extent relies on the photons that are emitted by the laser. And there's plenty of photons coming from the laser. Now, how does this work? Now, imagine now that you do not locate the molecule in the wide field mode, so with a plain illumination, the wide field illumination, but rather than using wide field illumination, we inject the position into the sample space with the zillions of photons that come from a laser. And how can we do that? A simple way of doing that is to convert a laser beam into a donut mode. And of course, the donut has a center, zero intensity center that is shown in here. And this position, of course, will be perfectly defined because it's done with zillions of photons coming from the laser. You can instantly check for that position because it's the position where there will never ever excitation photon arrive. So green is now excitation light, and then the center is where excitation photon, green photon never shows up. This position is perfectly known, as I said. Not only it is known, it can also be changed very easily, because here we have, in the scheme, we have a beam deflector, so something that can push that donut around, and hence also the center of the donut around, very easily, very precisely, with angstrom, picometer precision, if you like. And you always know where it is because you are in control of the position of that zero. So in that case, the whole localization process boils down to finding the position of finding the unknown, inherently unknown position of the molecule with respect to the known position of the donut zero. And how can we find that out? So how can we find out where the molecule is in relation to the known position of the donut zero? It's very simple. If the molecule is here, there's a lot of green light, then of course the molecule will emit fluorescence, and this will be measured at a detector here. Note, there is no camera. It's just a detector. The jargon is it's a confocal detector, meaning there's a pinhole and there's a simple photon counting device behind that pinhole. So counting the number of photons detected by seconds, by unit rate, unit time. Now, if there is green light, there will be a substantial detection rate, so the substantial number of detected photons. But if the two coincide in space, there won't be any detection in here because there's no excitation light, and therefore there is no emission and there's no detection. Yet we know where the molecule is because it must be exactly at the place where the donut zero is, and that position is known. So we know the position of the molecule in this case because it is the known position of the donut zero. So in other words, if the molecule is slightly away, then there will be some photon emission. But that photon emission will be much less than in the previous case. But as I said, to cut a long story short, the rate will tell us where the molecule is. The number of photons detected per unit time will tell us where the molecule is in relation to the donut zero. Now, why is this concept kind of, I should say, superior in most aspects, most physical aspects, than the previous concept? In order to make this, say, kind of easily understandable, and with my last name being Hell, I decided to introduce a demon. And this little demon, of course, knows something that we humans don't know. Moreover, the demon can act on the donut zero and can push that donut zero around in the sample space at will with maximum precision. Now let's assume the molecule starts moving like this. Now the demon, because it's a demon, can instantly, say within an attosecond, attoseconds have become very fashionable lately, say within an attosecond can push actually the donut zero right on top of the molecule, within an attosecond, just like that. And in that case, there would be, of course, no fluorescence emission, because the donut zero perfectly coincides with the position of the molecule. Yet the demon could perfectly trace the position of the molecule very, very precisely, so with, picometer or atometer, whatever, precision, because the two coincide in space and there is no fluorescence emission. So in other words, you can come up with a concept, with the thinking, that at least in this limit, this conceptual limit of a demon, doesn't require any fluorescence emission, yet gives you high precision, and strikingly doesn't even have a wavelength dependence. Now you will ask me, okay, this is a place where optics is really, really strong in the world, and I'm sure you teach your students, as I did at some point, if you have freely propagating waves, and if you do something with freely focused light, somehow it must scale with the wavelengths, what you get. It's true. The normal super resolution microscopy that got the prize in 2014, everything the resolution still scales with the wavelengths. The diffraction barrier is overcome, yes, but still the resolution that you get in palm, instead, and so on, still scales with the wavelengths. Here there's no wavelengths dependence. And of course, as a physicist, you think, how can that be? I mean, how is this counterintuitive? No, it's in retrospect, of course, it's very simple. I mean, what do we do here? We have a point, which is the molecule. The molecule is a point. And of course, you have the donut zero, which is essentially a point. It's a point. And what you do is you match 2 points. And if you match 2 points, where should the wavelength dependence come from? Why would it matter if the donut is made of green light, yellow light, red light, infrared light, X-rays, whatever, microwaves? I don't care. It's just a point. Of course, the donut in this particular case, if you're talking about fluorescence, must excite the molecule. So that's the only limitation. But there is no wavelength dependence in here. So it's showing this is really groundbreaking concept, not just something small. And then, of course, the dipole indentation problem is gone. If you just minimize the number of fluorescence emission, why will those matter? They won't matter. So in practice, of course, in this limiting concept, all these problems are sorted out. Now, all these problems are sorted out, and you can enter, so to speak, a new field with new potential. Now, in reality, there is no such thing as a demon. And of course, in reality, this concept will not work ideally. That's very clear. But you can come very close to the ideal situation, and by improving that technology, of course, you come closer and closer and closer and make a big leap compared to the current state-of-the-art localization. So we replaced, of course, the demon with a controller, and the controller, of course, counts the number of photons that are actually registered by the detector, and then acts accordingly on the beam deflector. So in other words, if the molecule starts moving now, the controller will, of course, count the photons. It's clear, because controller is not a demon. It cannot push the zero just on top of the molecule within an attosecond, of course. It will take some time. And so there will be signal coming. But the thing is, it's very clear, the closer the zero is on average, the molecule, the closer the demon situation is improved. So what these photons actually account for, these photons, of course, account for the residual distance between the zero and the molecule. And these photons, of course, are now, if you like, information science, more informative, because they will instantly tell you, so to speak, how far the molecule is away. Just a few photons tell you how far the molecule is away from the donut zero. Or another interesting physical picture is that you will, of course, as a physicist, if you're a physicist, instantly appreciate that if you want to have a very sharp expectation value for space with freely propagating light, photons, of course you need zillions of them, it's very clear. And that's why you need so many of them, of course, in the normal fluorescence localization. So what this concept does, actually, it takes away the burden of requiring many photons to get a sharp expectation value from the molecule. So it doesn't require the molecule to emit those photons, to provide those photons. It puts that burden on the laser, and the laser has plenty of photons. That's why this concept is so powerful. Basically, you need many photons to get a sharp expectation value, but you ask the laser to do it to a large extent, and so the photons that are still left for the molecule to emit are rather minor in number. So this is why this concept, I worked hard on coming up with a good acronym, so I eventually came up with this min-flux, which kind of alludes to the fact that you have minimum and you minimize the fluorescence flux rather than maximize the fluorescence flux. Well, I got stuck with it, and so it's called min-flux anyway. So in practice, if you want to have an equation that kind of gives you a feel for how precise we can be, then it's this one. The sigma is an uncertainty, so a precision, if you will. It still scales with the number of detected photons square root, but that's not important now because we have a handle. Here, the value of L, yeah, you can call it the average distance between the molecule and the donut zero, tells us how small we can make sigma in practice. Because if L goes down, if we just minimize the distance between the zero and the molecule, sigma goes down as well, meaning precision goes up. So we have a way of making sigma very small, even if we keep n constant. So in the previous case, of course, you had to increase n. So in order to get a 10 times smaller value of sigma, you had 100 times more photons. But here, you just make L 10 times smaller and you have it, yeah? And that's a new aspect. So in practice, the biologists say, okay, don't bore me with this physics, and I don't want to hear that. So how much do I gain? Yeah, well, so that's why I'm giving you a rule of thumb. It's about 100 times fewer detected photons that we need to get a certain precision as compared to a camera. But that's not a law, of course. This is just our current implementation, and the future will get better, and for some molecules better, for some samples it's better, for some samples it's slightly worse. But as a rule of thumb, just keep in mind. This has allowed us to demonstrate back in 16, 17, when we published this first, fast tracking, in this case of a ribosomal subunit. This is all unimportant. This was just the first evidence to prove that we can track faster than what people usually do with a camera. And this is just, I'm showing this picture here just to give credit to the very early work. So, but of course, early work and principle is nice. If you think about now as a physicist, and I'm just a physicist, I'm definitely not a biophysicist, and for sure not a medical physicist. If you think about it, what could you do? Well, keep in mind we can be 100 times faster because we need 100 times fewer photons. So when people usually require about 100 milliseconds to get a decent precision of localization, we require only 1 millisecond, or alternatively, we can be 10 times more precise. So then a standard localization with a camera. And so one obvious thing to do, you go for those that everyone thinks that everyone knows, including myself. Yeah, this is arguably the most famous, one of the most famous cartoons in science, the walking of kinesin on microtubules. It walks supposedly like this. Yeah, kind of human-like, don't you think so? It looks kind of suspicious or too good to be true. Well, I'm agnostic, so but this is how people draw it, and it's retreated even by, I don't want to mention the name. Anyway, so how was it found out? It was found out by pioneering experiments, really commendable experiments that I appreciate the groups who did it. The scientists are great scientists, and they really did pioneering work back in the 90s, for example. Steve Block, for example, and Karel Svoboda. What they did, they put a bead on the stalk and put it in an optical trap. As you may know, the optical trap actually got a physics Nobel a couple of years back to Art Ashkin. And this was one of the key applications, actually. But if you need a large bead, 200 nanometers or larger, to watch basically the tiny kinesin, which each of these feet or head or what you actually call them is 4 nanometers. It's a kind of thing. I mean, it can happen that you have 2 of them there. It's not so easy to control. And of course, it doesn't give you the direct movement of each of these motor domains or feet or head or whatever we like to call them. So, but of course, steps were seen for the first time. Now, Paul Selvin, about 10 years later, at that time, I think at Urbana-Champaign with Ahmed Yildiz, they decided to put, in famous experiments, they decided to put a fluorophore, which is much smaller. Fluorophore is just 1 nanometer, is light, no viscous drag, and so on, directly on the motor domain, so on the foot or head. And then, of course, watch how that fluorophore moves in space. And keep in mind, we're talking about localizing fluorophores, finding position of fluorophores. So this thing is just made for localization. Or you can put it on the stalk, as it is shown here. Then you watch the movement of the stalk, the spatial dislocation of the stalk. And then people say, well, fluorophores, it's still slow, and they're right, because with a camera, if you want to have 1 nanometer precision, you need about 350 milliseconds. He called it Fiona, fluorescence localization with 1 nanometer accuracy, precision, I should say. It's slow. And then people say, okay, let's put a gold bead on it. If you put a gold bead, there's a much higher rate. We don't need the fluorescence emission, which is low rate, then we're much faster. Experiments were done, good experiments, I don't play them down. But especially if you put a gold bead on a foot, then it becomes kind of dodgy, because you have a huge gold bead, 30 nanometer diameter, at the 4 nanometer sized foot or head, the motor domain, kind of, wow. I don't know. Anyway, so we have now the option of using MinFlux. So we can use Paul Selvin's approach of adding a fluorophore, a tiny fluorophore directly onto the foot or head or motor domain, or directly on the stalk, or on both. Okay? And we have the advantage now that we are faster, because we need 100 times fewer photons. We can see faster movements, or we can be more precise, whatever we want, actually, depending on how we want to use our photons. And of course, the best is that you label both feet, and then you see what's going on. Anyway, so we started out actually by labeling the stalk. So there's no load on it or some things, just stalk. And then, of course, if it moves like this, you anticipate it makes steps, so 16 nanometer movements of each of the feet or head or motor domains, and the stalk goes for 8 nanometers. We have seen those. The precision is high, and the spatial temporal resolution was indeed higher, much higher than in Paul Selvin's case, due to the fact that it's MinFlux. That's allowed us to see substeps, 4 nanometer substeps, that could not be seen under, say, no load conditions, or under say, just labeling conditions, and the precision of each step was below 1 nanometer. So we could see substeps and so on. So this clearly shows that the methodology of MinFlux gives you mechanistic insights that you couldn't get before. But not only that, Paul Selvin in those days, in those pioneering experiments, had to reduce the concentration of ATP by 100-fold. So the movement of the motor is driven by ATP. That's the energy currency of the cell. We could go to 1 millimolar ATP, 100 times concentration. Why? Because we're faster. So we can allow ATP to walk at its natural speed simply because we can track it. But he couldn't, yeah, but the camera is too slow and couldn't track it. And so we could go up to 1 millimolar ATP concentration. We could check out whether ATP is taken up when both feet are down or just one up in solution. I don't want to bore you with this. I would like to go to something that is more recent, where we labeled both feet. One with a green color, one with a red color. And then, of course, what you anticipate, that you see better now the movement of these individual motor domains individually. So you anticipate this kind of thing, yeah? It walks like this, as you've seen in the cartoon. But what we got is actually this. It's microtubules on a cover slip, just to be clear, microtubules on a cover slip, and the kinesin walks on top of the microtubules. Now, if you look at this trace, so this is here is time, this is on-axis position. Now let's look here at the center of this trace. Indeed, we have what we anticipate. Green moves first, then red passes, then green passes red, and then red passes green, and green passes red, and so on. It moves like this, humanized, this humanized fashion, as you've seen. But there's a funny thing. There's a big step in here, there's a big step in here, and if you look at the whole trace, yeah, it doesn't seem to walk like this, as it is shown here in the cartoon. But if you look at this part of the trace, you see, oh, it is kind of weird, because the red comes, and then the green just closes up to the red, and so on. So it moves more or less like this, making individual steps, 16 nanometer steps, but in this inchworm style. Now, if you go back to the literature, and you find that this was debated actually in the 90s, whether there is inchworm or this humanized hand over hand or foot over foot movement. And the inchworm was dismissed based on the fact that Paul Savin didn't see these big dislocations at the same time. And he anticipated 8 nanometer steps rather than 16 nanometer steps, because if you look at the functional units of microtubule, you would not anticipate 16 nanometers, but that's what we see. I mean, we've seen it, I can't help it, yeah. It's a fact. Also, this part shows the same thing, and we've seen it in 30 to 40% of the stepping cases. So it's not just rare. It surprised us as well. I didn't order it, you know, don't get me wrong. But there is this kind of large step that kind of means a passing step. So you can come up with a cartoon, and this is what the measurement is actually, that you have this kind of inchworm step, and then you have a large passing step, and then it starts the humanized walking style. And then again, you have a large passing step. There's also a debate whether it rotates around itself or just passes it like this. They call it symmetric or asymmetric. Currently, the notion is it's probably symmetric, but I cannot say at this point. Honestly, I don't know. I'm just telling you, this is just work in progress. And I also don't know if this is, so to speak, true in the cell. So this is all done in vitro. Just keep in mind, I'm not claiming anything, but this is what we see. Can we rule out optical artifacts? We can. So that I can say this is a true measurement. So, but we need to find out what it means. Okay, the next step was then to do it in a cell. And so I was fortunate that I had a talented student here actually, who was basically looking for a job because his wife got a job at the University of Mannheim and said, I want to do my PhD in Heidelberg and have a lab in Heidelberg. And so, okay, I find something for you. Why not looking into dynein? He managed actually to get dynein, endogenous dynein labeled in the living neuron through a CRISPR-Cas modification. And so these are the first recordings ever of moving dynein in a cell, full stop, yeah, in the living neuron. And you see them here, these are steps. Endogenous dynein is not overexpressed and this kind of stuff. So it's the real stuff. Now there is a literature, if you look at the literature, you will see that, oh, there's probably a tug of war between kinesin, dynein, retrograde, anterograde transport, and so on. We've seen those things, it is shown in here. But very rarely, 10% of the cases. What we see, and this was not anticipated, a quick reversal. It goes back and forth. It quickly decides to change its direction, not tug of war and then, oh, we have to go this way. So there must be a reversal mechanism there that is very effective, very quick, that makes up its mind and then changes direction. This is an observation. I don't know the mechanism. And I, as I said, I'm not a biologist, I'm not even biophysicist. It will be interesting to look into this, but one of my motivations is to kind of tell the dynein community, look, you've been tracking molecules with cameras, you've been attaching beads, you've been doing all kinds of things, all fine, all great, you got a lot of information, but now there's a new tool that can act like a game changer and can show you things that you couldn't see before, and I'm just scratching the surface of it here. There is something. There's something with kinesin, maybe it's not that humanized in its walking style as you may think. Maybe dynein doesn't have this tug of war at least not that much as you may think with kinesin. Maybe there's a quick reversal mechanism. This is what we see. So here, the advantage over the kinesin experiment, it's in the cell. It's in the living cell endogenous proteins. It just came out in PNAS, by the way, 3 days ago, so it's very hot. Anyway, I'm going to speak about resolution now, coming sort of towards the end of my talk. Now, you know, resolution has been my pet problem, and to some extent it still is. Now that MinFlux, as you've seen, has such a good localization precision, as I said, 10 times better than normal localization, meaning that in the past, if you had 10 nanometers, with the same number of photons, you get down to a nanometer. If you have 5 nanometers, the precision can even go down. I'm talking about precision, below a nanometer, can even go down to angstroms. So why not taking advantage of this? And one of the students in my lab, or actually a postdoc, said, there is this classical experiment which was done in the 60s, actually by Strickland Holland, when they want to prove the viability of so-called FRET, fluorescence resonant energy transfer for distance measurement. Now that we have such a high precision, why don't we measure molecular, intramolecular distances directly with MinFlux? Why not doing that? Why just taking pictures of large-scale stuff? And everyone does that by now, we cannot do that. And so actually we prepared a kind of ladder of prolines. So this is a multiple proline, so you see the number here, starting 5 out, going to 40, and there's 2 dyes at different ends. Now, as I said, MinFlux locates very precisely the position of each of the dyes, but that doesn't give you resolution. It just gives you the position. To resolve something, you need to be able to discern it, to tell it apart, to know that it's this one and not that one. And the fundamental mechanism, I'm repeating this, and I have done this many times, the fundamental mechanism of telling things apart in all the so-called super-resolution anoscopy, whatever like methods. Start out with that, Palm, Storm, Paint, SOPHY, you can, whatever acronym there may have come up, it all is based on on-off, meaning that you have 2 features that you would like to discern. You make sure, in standard microscopy, for example, but also in Palm and Storm, that this one is not emitting when this one is emitting, and this one is emitting when this is not emitting. This is the way you discern it. Doesn't give you the position, sure, but it allows you to see these are 2 different things. Okay, so what we implemented here is an on-off transition. So meaning we made sure that when this fluorophore was emitting at some point, this one was clearly off. And at the time this one was emitting on the right-hand side, the one on the left-hand side was off. So this on-off mechanism is required. And we did that, of course, with a fluorophore that had a very strong orthogonality, meaning that when it was on and emitting. But it was never off, more or less. And when the one that was off was always off, So it was not on by coincidence. So I would like to put this still in relation with FRET, because this is kind of interesting. So FRET has been used actually for decades, still being used, still has advantages. I'm not playing it down. So, but FRET works like this. For those of you who don't know, you have 2 fluorophores, one that is kind of bluer than the other one. So blue shifted with respect to the other one, and then if we excite the blue shifted one, it gives part of its energy to the acceptor, and the acceptor emits, and you can infer actually from the transfer of energy how far they are apart. Then you know the distance. Now, since these are dipole transitions, if they are oriented like this, there is no dipole interactions because the dipole are mutually orthogonal, then you cannot measure. And so this orientation factor is really a nuisance because it kind of compromises severely the distance. The specialists will call it the kappa square factor, so the orientational factor. And not only that, that you get wrong distance because the efficiency of transfer is compromised. It's also inaccurate to some extent, or it can be inaccurate because you have a strong dependence, of course, here on the 6 power. That also kind of makes it to some extent dodgy. You have also dependence on the local environment, which makes it inaccurate. But this strong dependence on the 6 power limits limits actually the viability range to 2 to 8 nanometers. So if it's larger than 8 nanometers, the transfer is too weak. If it's too small, then there is also issues coming up, which I don't want to mention it's here in detail. So you are limited. But the on-off, say, separation, of course, if once you've implemented that orthogonally, such that one is off and the other one is on, and never vice versa, then you can have perfect separation. And in combination with this high precision, you should be able to separate intramolecular distance. So we did this experiment again, and you see now this is 3 to 11 nanometers, and this is the Fred range, and of course we can go beyond the Fred range, so there's no problem. Why not separating 10 nanometers, 12 nanometers, 20 nanometers, 15 nanometers, there's no problem. So by direct imaging. So you see now measured distances, 2.8 nanometers in this case, 3 angstrom precision by direct imaging. Now this is an interesting thing. I prepared this actually for White, because White group actually, to my knowledge, also worked with Christian Griesinger when they proved actually the viability of the cold concept, where large numbers of photons were generated with by cooling things down. Definitely works, definitely gives the very high precision. He got it first, got down to very, very high precision. But in this case, of course, since we spare the number of emissions, since we don't have to do this, we don't have to, of course, go to low temperature. We can do it straight in under, say, liquid conditions. Now, 11 angstrom distance here. This is a bacterial citrate sensor, actually, that Christian Griesinger and Stefan Becker use for particular reasons. It's not interesting why. So here the distance is known, 2 molecules. We played on-off, so they were sequentially emissive, and then, clearly vindicated. 11 or 10.9 angstrom distance, 0.6 angstrom precision. You won't be able to go below 1 nanometer because the size of the fluorophore is 1 nanometer. So they can basically touch each other, but then you're done. Yeah, so can you have a smaller distance. But in projection, of course, if you do a 3D imaging, the distance will always remain the 1 nanometer, but the projected distance, of course, can be smaller. And here it's measured 6.6, 3.1. Basically, it goes down, well, to nothing. So in other words, you're kind of in control of dimensions, despite the fact that's all done with focused visible light, room temperature, aqueous conditions. And so this is kind of weird, especially if I'm sure you remember the 20th century, not all of you, of course, but some of you will remember the 20th century. So when I studied and you studied, no way of getting anywhere far below 200 nanometers. Now we are controlling distances, say, of 1 nanometer, and to some extent even sub-nanometer with optics. The square root of n method has been used by Jungmann et al. As some of you may know, by getting a high number of emissions, a high number of photons emission by reattaching fluorophores that eventually bleach and then they reattach and emit. But that method still requires large number of photons. And in my view, it still doesn't sort out the problem of the orientation. So if you ask me, the way to go, what is the way to go to those small scales in proteins? It is the direct labeling. Because you have a small label, you don't have any mediator or something like that. And the orientational problem is largely mitigated in this case. So I said this on-off thing many times. Actually, I said it, believe it or not, in the banquet speech at 2014 Nobel banquet. So banquet means that there is the king, there is diplomats, Randy will know, it's not just scientists. Still, I put this on-off as the key discovery for the 2014 Nobel Prize. And I've said this repeatedly, and those who know me know that I've said this basically in every talk, and if you say it many hundreds of times, then you sound like a broken record, and at some point you ask yourself, maybe I'm wrong, maybe somebody comes and proves me wrong, and then this doesn't feel nice, so better I prove myself wrong. So I started to think about, is it really needed that we do on-off? Yeah? Is that really fundamentally needed? And the answer is no, and I show you why. Let's revisit Rayleigh's criterion. I think everyone who has a physics background knows Rayleigh's criterion. You have two things that are closer than diffraction barrier. You cannot tell them apart unless you play on-off. But we don't play on-off. We just keep everything on. You have two molecules. Let's assume for the moment this sort of thought experiment where you have the two molecules sitting very close, and then we put them into a microscope. Let's assume it's a confocal microscope that uses a Gaussian beam, or so a regularly focused beam. The two molecules are too close to be separated. What do you get? You get an image that looks like this, okay, like a diffracted image that looks like a Gaussian. In fact, it's hard to discern from a real Gaussian, because if the molecules were at the same position as they are here, yeah, you would get basically the same image as you do get here if they're slightly apart. So the difference between this plane Gaussian, where the two molecules sit in the same plane, compared to where the two molecules are apart, is not visible. To be precise, it is flooded in noise. Because if you subtract the two, what you get is noise that you cannot really interpretate. If there was no noise, if those images were analytical, of course, completely analytical, then of course you could separate, you can deconvolve, so to speak, and calculate back. That's pure mathematical, simple mathematical problem, but there's no such thing as noise-free signal. We do, after all, measurements. Definitely with fluorescence, there is noise, believe me. Anyway, so if we subtracted those profiles, yeah, here, we subtract those profiles. So this is where the two sit in the same place. The orange is where they are slightly apart. The difference between the dark one and the orange one is completely within the noise band. So you cannot separate. That's why people said, okay, of course, you cannot separate it. Let's stop talking about it. It's very obvious that you can separate it. But let's do the same thing with a donut. Okay, now let's assume for the moment we scan with a donut beam, okay? Like in MinFlux, we do MinFlux, but we don't play on-off. The molecules are on, and we scan with a donut beam. So we have a donut beam. The molecules sit in the same place. If we scan the donut beam across the molecules, what will happen? We'll get an image that will look like a donut, and with a donut that has a true zero at the center. Very clear, because if the two molecules coincide with the position of the donut center, there will be no signal. True zero. But what happens if the two are slightly apart? Well, of course we get a donut-like image here, but for sure the zero here at the center will not be zero. It can't be zero, because when one molecule is at a zero, the next one won't be, because they are slightly offset. So it's impossible to get a zero signal here, yeah, with two offset molecules. So in other words, we can clearly discern those situations. In fact, if you subtract the two, unlike here you get a signal that sets itself apart from noise, from background, because background is zero. Or in other words, you can say, with physicists, we learn never measure against background, measure against zero signal. And what we do here is we measure against zero signal here at the donut center. Here, we measure the signal of one molecule on the backdrop of the noise of the other molecule. That's why it doesn't work. But here, clearly there is discernible signal. So in other words, you can separate them. If you scan with a donut, you can separate them, the molecules, even if they're close. And the crazy thing is, in the absence of background, in fact, the closer they get, the better you can separate. Kind of weird, yeah? So what does this mean? Means that we should be able, also in reality, if the concept is so strong, and obviously it looks like a very strong concept, you must be able to separate two molecules, even if they're far below the diffraction, especially if they're far below diffraction barrier, even if they're on all the time. So here's the measurement. So 35 nanometers, 25, 20, 15, 10, so 8 nanometers, and here is the separation. For example, here those molecules were on both at the same time, slightly below 9 nanometers, clearly separated. You can separate them. So that's clearly possible. And the good thing about it, it doesn't work only just for two, it also works for more. But why is this important? It's very simple. If you need to play on-off, as you need to do instead, you need to do in Palm, you need to Storm, you need to do in Paint, and the rest of it, Sophie, whatever you want to call them, in the whole 2014 Nobel gamut of techniques, it's impossible to track them at the same time, because if this is off, of course you cannot see it. It doesn't know what it does. Yeah? Only this one is on. Yeah? So you cannot observe it if it's off. So if you need to play on-off, there is no such thing as simultaneous observation. But if they are on, all the time on, then you can observe them constantly. So here you see on the left-hand side, yeah, you see two molecules 15 nanometers apart, yeah, tracked all the time. It means you can track and separate them at the same time. And that's arguably unbelievable, but it's true. And this brings me actually to a new vision. And that vision is actually something that really kind of keeps me up at night, as Americans would say. We have now very good techniques, I should say, for knowing the structure of a protein. Yeah, we have X-rays, we have NMR, cryo-EM, great technique, and even AlphaFold. They predict within a short period of time the structure. But there is hardly any technique that I know of that really kind of instantly shows you function at small scale. So how does the protein actually change its shape, change its conformation, does something directly measured on a single protein scale? Now we could do the following experiments. Since we don't need the on-off, so we can track fluorophores constantly all the time. We label maybe 4 or 5 or 6 or 7 or 8 or 9 pieces with the same dye. Very good dyes, need not be switchable, need not be photoactivatable. These are bad dyes, by the way, they are very lipophilic. Very good dyes, the best that you can imagine, bright, constantly emitting all the time, and just track them on small scales, and we would know how the protein works. Put a drug, see if the drug changes, how it works. So I think this is really exciting, and I believe this is the way to go, and it's most likely opening up a new field. Then there's another vision that's more kind of physicist thing. So what do you think? Why was the diffraction barrier overcome for fluorescence microscopy and not for other techniques? So when I looked into this problem first in the late 80s, a kind of a crazy maverick PhD student at that time, which I definitely was, definitely the maverick, I wanted to overcome the diffraction barrier for anything, of course. And of course, not for fluorescence microscopy in particular, definitely not. Honestly, I admit it now because, I think it's kind of acceptable. In those days, I was so arrogant as a physics student, I would never, ever have done anything for biologists. Because biologists were those guys who learn things by heart, and so I never do something for biologists, of course. But I finally ended up with fluorescence because I realized that's where you can overcome the diffraction barrier. Why? Because you can take advantage of the fact that fluorescence is a phenomenon that you can easily manipulate. Fluorescent molecules have states that you can easily modify. And so you can play on-off, and that was the key idea behind that, of course. So that was essential. But now, if you don't need on-off anymore, it means there is no fundamental need for fluorescence. The advantage here of fluorescence, why did I show is fluorescence? Because, it gives very little background, it's very easy, but there is no fundamental need for fluorescence. So this concept I could have shown with Raman scattering and the rest of it. Yeah, there is no fundamental restriction to fluorescence. So what I'm saying as a kind of, summary at the end, I think this concept is not just another concept for super resolution, what people would like to call it. I hate this word super resolution, by the way, but it has become fashionable, and so I have no choice but using it. I would like to, if you don't care about all this stuff that I'm showing, just keep in mind this thing. So people who build cars, they said, okay, my car has 4 cylinders, I want something more fancy, I put 6 or 8 or 16 cylinders. Then I get a higher torque and a higher acceleration. But it doesn't really scale, as you know, It's the same with the camera localization. It scales with the square root of n, so you need 100 times more photons to get 10 times higher precision, and 10,000 more photons to get 100 times. Doesn't scale nicely. It's not nice physics behind it, apart from the orientational problem. So this is the old way of doing it. You can do away with cylinders, take an electric engine, You may like it or not. I mean, Audi is not far from here, and I'm in a state where really that is in deep trouble now, Volkswagen. I'm in Niedersachsen, Lower Saxony. So I know what I'm talking about. But anyway, 0 cylinders, of course, high torque, high acceleration. Jochen, I think this resonates with you. And this is what MinFlux is about. And then, keep in mind, the precision scales exponentially with the number of photons. This is not with the sluggish, bad square root function, which is slow, square root of, I don't know what, is just nothing. It scales exponentially. Why does it scale exponentially? I'll give you a hand-waving argument. Of course, when it comes to finding out something, you want to find out within the diffraction barrier. Diffraction barrier is 200 nanometers. So let's assume we have something within this 200 nanometers, where we don't know where it is. So you need one photon to know if it's in this part of it, in this half, or in that half. So you have one photon, you know it's here. Then you have another photon, and it's here. Then you have another photon to know it's here, another photon, it's in here. You can figure out that with 20 photons, you're down at a nanometer. Of course, it won't work that idealistic, as I'm saying, but this shows you that this concept is a game changer. It's not just a new variant or so, it is a true game changer. So with that, I'm thanking you for listening so patiently, and I'm thanking a small but very, very, very talented group of people that I work with. Half of them are located in Göttingen, half of them in Heidelberg. You might think we are very much dislocated. Yes, we are. We have at least some basic quantum mechanics experience, but we have a lot of fun pushing this field, especially now that it seems to open completely new avenues. Thank you very much.